In this video, we demonstrate the software functionality of the Atomic Force Microscope setup developed in the lab. There are 5 software steps to begin an imaging process. First, we start the LabVIEW software by clicking on the white arrow. Second, we initialize the scanner position by click on Move to Start button using the preset imaging parameters. Third, we start to drive the probe and observe the amplitude output from a custom-designed lock-in amplifier. Fourth, we engage the probe until the tapping amplitude reaches a threshold. Fifth, we can now begin the imaging process and watch the 2D displays for topography and deflection update. We can observe the optical view of the probe and sample as shown on the screen. A 3D display can also be turned on at this imaging rate for better visualization. In this video, the sample is a calibration grating with 500 nanometer depth, and 8 micron measured pitch. The imaging is conducted in tapping mode with a 20 by 20 micron range, at 400 by 400 pixel resolution, with a line rate of 2 lines per second. Imaging data are recorded automatically for post-processing. After the image is taken, we can simply click on the Stop Imaging button, retract the probe and stop the software. Better visualization of the image can be obtained through post-processing in MATLAB software as shown in the animation. Thank you for watching.